Good morning, students. Today we are going to discuss on the topic gametogenesis. Okay, gametogenesis, the name, it combines with two words, that is gametes and genesis. This gametes means what? Can anybody tell me what are the gametes? So gametes are the germ cells. They are involved in the process of reproduction to produce new individuals. Okay, so this process of gametogenesis which are taking place in the gonads. So here we should know the gametes and gonads, the difference between gametes and gonads. The gametes are the germ cells, already I told, that is spermatozoa in male, ova in the female, and gonads are the female gonad is ovary and the male gonad is testes. So this process of gamete formation, genesis means formation, gametes means germ cells. So these gametes formation takes place in the gonads. So this process, the process of formation of gametes within the gonads is called as gametogenesis. Okay, this is clear. Then so we already I told you testis is the male gamete and the ovary is the female gamete, as you are seeing in the slide. So the process of gametogenesis which occurs in two types. So gametes already I told male and female. So that's why there are two types in gametogenesis. That is, one is spermatogenesis and another one is the oogenesis. So spermatogenesis which is taking place in the males and oogenesis which is taking place in the where in the females. Okay, next we should move to the second slide. Next, so first one is the spermatogenesis. So spermatogenesis process taking place in the males, only in the males, not in the females. So in the females there is another process, that is a oogenesis. So spermatogenesis means, so here I told the gametogenesis means process of formation of gametes. Here spermatogenesis means the process of formation and maturation of sperms in the testes is called as spermatogenesis. Under the process of sperm formation and maturation, the process of formation and maturation of sperms in the testes, where it is taking place in the testes, that is male gonad, is called as spermatogenesis. So this is clear. So the process of spermatogenesis involves two main steps. Means the process of spermatogenesis completely we are going to study in the two steps, two major steps. So one is formation of spermatids and another one is the second step is the spermiogenesis. What are those steps? First one is the formation of spermatids and second one is the spermiogenesis. So these two steps you have to remember. Next, so first step is formation of spermatids. So the name itself indicates formation of what? Spermatids. So here the formation of spermatid is a process. So it is one of the process. So in this process what happens is, during which the primordial germ cells transforms itself into spermatid. So here what happens? It is one of the process. In this process, the primary germ cells or the primordial germ cells we can call. So these cells transforms themselves or itself into spermatids. So the primordial germ cells they transform into themselves into the spermatids. Now this is the formation. So the process is further differentiated into three phases. So this formation of spermatid undergoes three main processes. So those processes are, or those phases are, phase of multiplication, the phase of growth and phase of maturation. So these three phases which are involved in the process of formation of spermatids. So one by one we can see now in the next slide. Okay, the first one is phase of multiplication. So here multiplication we are studying the phase of multiplication phase. Multiplication is multiplying the cells. So during this phase, some of the germinal cells or the germ cells divide repeatedly. The germ cells or primordial germ cells, they go on dividing repeatedly by the process of what? Which process? That is mitosis process. So the mitotic division, the primordial germ cells undergo the mitotic division repeatedly, and they produce large number of cells, that is diploid daughter cells, called as spermatogonia. So this diploid daughter cells are called as spermatogonia or the spermatogonial cells. So some of these spermatogonia continue as stem cells or type B spermatogonial cells and continue to divide. Means what happens? Again these spermatogonial cells undergo mitotic division to continue for the division process, while the other differentiate into type B spermatogonia, which are precursors of the sperms. In the next slide, in the upcoming slide, I will show the schematic representation of the spermatogenesis process. So in that diagram you can clearly understand what the process is, and it is easy to remember the process. Okay, so you just remember the schematic representation, and you should draw the diagram or schematic representation, and you can easily explain it the examination. Okay, so type B continued spermatogonia, and this enters into the next phase called as phase of growth. So some type B spermatogonial cells, they enter into the next phase. That is next phase is, which phase is the next phase? That is phase of growth. Okay, so here the cells are going to be multiplied. Multiply ಆಗ್ತಾ ಇರ್ತಾವೆ ಈ ಸೆಲ್ನಾಗ. ಇದು ಒಂದು ಫೇಸ್ನಾಗ ಸೆಲ್ಸು ಮಲ್ಟಿಪ್ಲೈ ಆಗ್ತಾ ಇರ್ತಾವೆ. ಸೊ ಮಲ್ಟಿಪ್ಲೈ ಆದಮೇಲೆ ಏನಾಗ್ತಾವೆ? ಸೊ ದೇ ಗೋ ಟು ದಿ ನೆಕ್ಸ್ಟ್ phase, that is called a phase of growth. So that we can see in the next slide. Okay, next. So phase of growth. Growth means here the cells are undergo the process of growth. So during this phase, the diploid type of B spermatogonial cells undergo growth. So the phase multiply ಆಗ್ತವೆ, growth ಆಗ್ತವೆ. That only you have to explain. So undergo growth for a brief period and almost becomes a double in size. So the growth of the spermatogonial cells takes place. These enlarged cells are called as primary spermatocyte. So that enlarged one, the enlarged cells are called as primary spermatocytes ಅಂತ ಕರಿತೀವಿ. So which are still diploid in nature. They have not become haploid. They are still in the diploid state. So these cells enter into the next phase, that is called as phase of maturation. So here only the growth of spermatogonial cells takes place, and the cells which have grown, so those are called as primary spermatocyte, and it is still in the diploid stage. Diploid state or diploid in nature. After growth, what happens? The sperms has to be mature, or the cells has to be mature. So these primary spermatocyte next enter into the next phase called as phase of maturation. So the third stage in the formation of spermatid, that is phase of maturation. So during this phase, primary spermatocyte undergoes meiotic division. First what happened? In the first phase, multiplication phase, the primordial germ cells go on dividing repeatedly by the process of mitosis. But here in the phase of maturation, here there is no mitotic division taking place, but instead of mitosis, meiotic division is taking place. Here the primary spermatocyte undergoes meiotic division. So at the time of first meiotic division. There are two meiotic divisions taking place at the time of first meiotic division, which is a reductional division. That is, the homologous chromosomes undergo synapsis, synapsis, and the crossing over resulting in the genetic recombination. So here the meiotic already we have studied in the previous class. That is in the means in the PUC also you have studied the gametogenesis also, and the division, cell division. In the cell division chapter, I think you have come across this meiotic and mitotic divisions, isn't it? So here the meiotic first division taking place, that is homologous chromosomes undergo this synapsis, and crossing over takes place and resulting in the genetic recombination. Okay, so at the end of first meiotic division, the diploid primary spermatocytes, the diploid primary, already they are in the diploid state, so those diploid primary spermatocytes divides into two similar haploid secondary spermatocytes. Second two ಅಂದರೆ ಮೀನ್ಸ್ ಡಿಪ್ಲಾಯ್ಡ್ ಇದ್ದಿದ್ದು ಏನಾಗ್ತದೆ ಇಲ್ಲಿ ಹ್ಯಾಪ್ಲಾಯ್ಡ್ ಆಗ್ತದೆ, ಯಾಕಂದರೆ ಮಿಯೋಟಿಕ್ ಡಿವಿಷನ್, ಬಿಕಾಸ್ ಇಟ್ ಈಸ್ ಅ ರಿಡಕ್ಷನಲ್ ಡಿವಿಷನ್, ರೆಡ್ಯೂಸ್ ಆಗ್ತಾ ಇರ್ತದೆ ಇಲ್ಲಿ. ಹ್ಯಾಪ್ಲಾಯ್ಡ್ ಸೆಕೆಂಡ್ರಿ ಸ್ಪರ್ಮಟೋಸೈಟ್ಸ್, ಸೊ ದೋಸ್ ಆರ್ ಕಾಲ್ಡ್ ಆಸ್ secondary spermatocytes which are haploid in nature. Diploid in the primary spermatocyte will be diploid in nature, but the haploid the secondary spermatocytes are haploid in nature. Then the secondary spermatocytes enter into the second meiotic division. The primary spermatocytes or the primary diploid spermatocytes undergo first meiotic division, but the secondary haploid spermatocytes undergo which division? Second meiotic division. Second meiotic division, which is equational division we can call, that producing 4 haploid spermatids at the end. So secondary mitotic division, what happens? This is a equational division. So these produce 4 haploid spermatids. Thus each primary spermatocyte gives rise to 4 haploid spermatids at the end. So this is all the 3 phases which are going to form the spermatids. So this is the complete 3 phases of spermatids. So the another one, that is, this is a schematic representation. So here you can see the first is the spermatogenesis process in the heading, and there is a spermatogonium, means primary germ cells or primordial germ cells. Then what happens? Those primordial germ cells undergo mitotic division to produce primary spermatocytes, and these primary spermatocytes undergo meiotic 1 division, and they produce what? Haploid spermatocytes. Those are called as secondary spermatocyte, and these secondary spermatocytes again undergo meiotic 2 division, that is equational division, and to form 4 haploid spermatids. So up to this is the formation of spermatids. And next one is the spermiogenesis. Spermiogenesis means from the spermatids the mature sperms are produced, and the sperms are going to be mature, the maturation process. You can see in the next slide. So here spermiogenesis. So spermiogenesis is the process of transformation of non-motile, inactive and non-functional spermatids into the motile, active and functional sperms. So is called as spermiogenesis. Spermiogenesis. So spermiogenesis already it is a process, and the spermiogenesis is also a process that will convert the non-functional, inactive, non-motile spermatids. Spermatids are inactive and they are non-motile. They are non-motile, they cannot move. And non-functional, they do not function anything. So non-functional, inactive, non-motile forms of spermatids are converted into motile, means sperms can't move, and they become active and they become functional. So this conversion or transformation of non-motile, inactive, non-functional spermatids into the active, functional, motile sperms is called as spermiogenesis. Okay, so during this process or this phase, what happens is the spermatids undergo a series of morphological and physiological changes. So the spermatids undergo a series of changes. So what are those changes? They may be morphological changes, means in the structure there are changes taking place, and physiological changes. In physiology also there is a, means the functional related also the changes takes place, and they can transform themselves into the functional sperms. So during this process of transformation, they anchor themselves to Sertoli cells and derive nourishment from them. What are Sertoli cells? The Sertoli cells give these are the cells, and they can give or provide nourishment to the growing spermatozoa. Spermatozoa has to go and they have to mature, so that's why they need some amount of energy, so some amount of nutrition. So that's why the Sertoli cells give the nourishment to the maturing spermatozoa. So once the process is complete, so they release themselves from Sertoli cells and enter into the lumen of seminiferous tubules. So seminiferous tubules andre ಏನು? Seminiferous tubules are the structure line functional unit of the testes. They are present in the testes. In the seminiferous tubules only the Sertoli cells and the after the completion of this process, what happens? The Sertoli cells and enter into the lumen of the seminiferous tubules. And they can release from the Sertoli cells after getting nourishment from the Sertoli cells, and they enter into the lumen of the seminiferous tubule. So this process is called as spermiation and spermiation. So this is about the spermatogenesis process. So in the last slide we have already observed, that is a schematic representation. Once again you can see that the representation. So up to spermatids you have seen, that is 4 haploid spermatids are there, and these 4 haploid spermatids are again they are non-functional, inactive and non-motile. So from those non-motile, inactive forms, they become mature sperm cells, that is the spermatozoa, by getting the nourishment from the Sertoli cells, and they undergo many changes, that is physiological, morphological changes, and become active, motile and functional. So this is the complete information about the spermatogenesis.